I would like first to thank the organizers for the opportunity of talking to you today about the Latin American experience, how we are building capacity locally and working within collaborative networks for single-cell analysis. So I guess by now we all appreciate the mission of the Human Cell Atlas, which is to create a comprehensive reference map of all types and of cells and properties of all human cells, with the future purpose of helping the understanding, diagnosing, monitoring, and treating diseases. The Human Cell Atlas has quite a few principles and values that they follow, and I would like to highlight equity, diversity, and inclusion, both of research subjects and scientists, which is very important to us today. Back in 2019, the Equity Working Group, co-chaired by Alex, Part, and Musa, which you will all be meeting by the end of this event, they realized that there was a very important underrepresentation of Latin America and Africa. And the question was, why? So to answer that, they put together two very interesting meetings led by the Equity and Diversity Working Group in London and Addis Ababa in the same year. And the idea was to put together very different people with very different backgrounds and expertise, and discuss the differences between equality and equity and how we could achieve equity within the consortium. By then, me and others, we had really never heard much about the Human Cell Atlas, so we understood that the first step was to create awareness about the consortium. So we got together to put together a first roadshow of the HCA in Brazil. And the idea was to present the initiative to Brazilian researchers and students. And we focused in São Paulo area, which is one of the regions in Brazil that gathers together most of the research in the country. So we thought it would be a very good beginning. The idea was to have lectures by the Equity Working Group, show results, ideas, how they intended to work with single-cell analysis, the kind of results that they could bring. And also, we also had the presence of funders who were represented by the Chan Zuckerberg Initiative, who had a lot of experience in funding HCA projects. From that, we moved on to having a real brainstorming with local researchers about how to apply to such proposals. And at the time, there was a call opened, and we got researchers together to discuss and to really learn about the technology, data analysis, and how they could collaborate in a proposal like that. That was a breakthrough for Brazil. So for the first time, CZI got a lot of research proposals from the country. So for us, that was a very successful way of going about. So after that, we got together with the executive committee from the HCA to put together the first HCA meeting dedicated to Latin America. Still, the organizers were mostly Brazilians because that's how it started, and we had to start somewhere. So that's why it started like that. And the idea of this meeting was then not to be focused in Brazil anymore, but actually to introduce the Human Cell Atlas to the whole Latin American community through the presentation of HCA regional networks, so people could see how it would work for us to work together. The HCA working group, so people could understand how the consortium was structured. And we were able to find 7 presenters from Latin America who hadn't really generated much data yet, but who were really interested in working with that and had some results to present based mostly on publicly available data. And we had over 400 attendees from 21 countries. So for us, this was a very important moment of integration from between Latin America and the rest of the community already working with single-cell analysis. One very important aspect of this meeting were the breakout sessions, during which researchers got together by affinity, so common interests, and those were researchers from all over the world, not just Latin America, and they could share ideas and ways to move forward, like what could we do, right, to start working with single-cell analysis and research in general, right? So within one of these breakout sessions, I got to meet very interested colleagues in putting together the first aspect of single cell research that we can actually make happen in Latin America, which is data analysis. So we organized a first single cell RNA-seq data analysis workshop in Latin America, this time organized by Mexico, Brazil, and Chile. And it was a very ambitious project. We were very happy that the HCA team supported us and believed in us. We had the first week of lectures. So the purpose was to instruct researchers and students on how to design single-cell RNA-seq experiments, manage and analyze data. So we had HCA members now from the bio-networks to present, and a lot of data analysis experts presenting their tools and what they were currently doing. The second week was a hands-on workshop using real single-cell RNA-seq data, not generating in Latin America because we don't have that yet, but which were publicly available. And the workshop was carried out, so hands-on in 3 different countries simultaneously, Mexico, Chile, and Brazil, and they were coordinated by local researchers. And at the end of the day, so that was a 5-day workshop, everybody would get together and discuss the tools they used, how they managed the difficult issues, the successes, and the characteristics of the results that they were getting. So that was a very fulfilling experience we had, and we are planning on having a second one next year. As real results from this collaboration effort in data analysis, we presented an abstract in the HCA general meeting last year with the results of the workshop. So co-authors were students that were present in the group. So that was very, very interesting for everybody and very rewarding. We also presented an abstract at the Genetics National Congress in Brazil also last year. And we have a book chapter that we put together based on the lessons learned also from this workshop on data analysis. But possibly our biggest success so far is to have been funded by CZR for the ancestry networks for the Human Cell Atlas. Our project is called A Human Cell Map of Latin American Diversity, and our team is made of Latin Americans, basically. So Mexico, Colombia, Brazil, Peru, and Chile, and US Latinos from Florida. We are happy that all the PIs are from Latin America, and all the expertises that we need are represented. We have community engagement experts in sampling from remote locations, sample processing, cellular biology, data analysis, and bioinformatics, and also training. We will, of course, collaborate with all the networks and with researchers from all over the world. We're all excited about that, but we're also very happy to have been able to put together a team that locally can perform all the experiments and all that is necessary to get the project going. The background for the project is the unique diversity of Latin America. So it's very heterogeneous. We have very local indigenous communities still present, but also a lot of admixture from indigenous populations and other continental sources. And these ancestries, they are underrepresented in current research in general, and we believe they may harbor undiscovered population-specific expression patterns at the cellular level. Here on the right, we have 2 examples of that. So this is ancestry results based on SNP arrays. And here, one of the ancestries we're interested in Chile, which is the Mapuche ancestry, and it's the effect of Mapuche ancestry on gallbladder cancer in Chile, which is very striking. So the goals of the project, single-cell RNA profiling of immune cell types across Latin American populations, both Native American populations and also mixed populations from Latin America. Create a new HCA bionetwork focused in gallbladder from Aymara and Mapuche ethnic groups from Chile. And also very important to us is to create a Latin American community portal for single-cell information, data sharing, and visualization, which we intend to make available also in local languages to help the inclusion and access by not only researchers and students, but also by the population in general. Just also as an example of how we're going to work, and we believe this is going to be very successful, we're going to have local processing hubs for samples in Mexico, Chile, and Brazil. So here on the left is a description of the areas we're going to be targeting, but processing is going to be in 3 hubs, and sequencing is going to be in 1 hub and genotyping in another hub. So a hub in Brazil and 1 hub in Mexico. That helps us cut costs, right? And not making it not necessary to invest so much in equipment. We're going to have joint data analysis with team in each one of the countries, and then training and return of results going to be discussed between the teams. What we hope to address with this project, so bias in data sets, improve representation of Latin American diversity within HCA results, bias in leading teams, so improve diversity of researchers spearheading efforts on their own populations. Single-cell RNA sequencing is mostly unavailable in Latin America these days, so this project will build local capacity, and then training. We'll provide basic and advanced training for Latin American students and also researchers, and in local languages, which this is a very important issue for us, and create this portal for data analysis and sharing, to increase access and inclusion and the understanding of the results from our project also within this data portal for the population. So we believe that we came a long way since 2019 up to now in creating a local community at least aware and engaged in what the Human Cell Atlas is doing, and possibly in the near future really generating a lot of data to contribute to the vision and mission of the HCA. We also believe we have come a long way to understanding what equity is. So mostly customized tools, solutions, framework, discussing with the community what the community really needs in order to make this happen. We are happy. We think we're moving in that direction. And I personally believe this kind of consortium is a step towards what we can really call justice, which would be a step further from equity, which means changing the system and offering equal access to opportunities to everyone. And with that, I thank you. And we are, of course, open to discussing our experience and sharing results. We have a lot of ancestry background that overlaps with the African continent. So I believe we're going to have a lot to do together in the near future. Thank you.
I want to use this opportunity on behalf of my colleagues and I to thank the organizers for this marvelous opportunity to talk about our ambitious drive to generate the first placenta human cell, Nigerian placenta human cell atlas from Nigeria. I'll be speaking, we arranged the slides in a way that I'll be speaking and Musa would also take over at a certain point so that we can both tell different aspects of the story of how this ambitious and daring journey began. So the outline of the slides, we'll be talking about who we are, how we met, the structure of our collaboration and project, and challenges and our future goals in general. We are 3 different scientists. We're on totally different, would I say, trajectories, career trajectories. And I'm currently based at Ahmadu Bello University. Just came back from the NHGRI fellowship. Musa is also currently based at Kaduna State University and in Nigeria, and Benedict is still available at the National Institute of Environmental Health and Sciences. And Benedict is actually a computational biologist. I have a specialization and a career trajectory on cell biology, while Musa is actually a community health and epidemiological physician. Initially, in 2019, The APTI fellowship actually moved Musa and I to the National Institutes of Health, where I was working with Francis Collins under the mentorship of Francis Collins at the National Human Genome Research Institute, and picked interest in single cell research under his mentorship. Musa, on the other hand, is working with Stephanie London at the National Institute of Environmental Health Services, where he met Dr. Anchang Benedict, who was at that time and still now the Stadman principal investigator and an expert on computational biology. So based on our shared interests of trying to actually translate scientific excellence and capacity building opportunities to Africa and attempting to change the paradigm of research methods in Africa, we started a conversation on a collaboration. And then the CZI Ancestry Human Cell Atlas core network presented a unique opportunity between us, and we did exploited our individual strengths and worked on different aspects of the proposal and pulled it together and decided to actually embark on this, to request for funding to embark on this, our very ambitious thought of building the first Nigerian placental cell atlas from a diverse set of women. Our team at first sight does not look diverse because we're basically Africans or from African ancestry. However, what we did in trying to build the proposal together was we structured it in a way that we generated a system for equitable staff recruitment under each independent principal investigator on this project. And so under my staff, I have predominantly female staff working with me, similarly with Musa and Dr. Anchang. And we try to make sure that the diversity is balanced in terms of ethnicity and different cultures, even though we probably may still be from the same African ancestry, but we'll make sure the ethnicities are actually diverse and the gender is well balanced. So in building the project, like I mentioned initially, we built up on our different strengths, and Musa being an epidemiologist and a community health expert, worked on the community engagement plan on how to eventually recruit and enlighten the community about the project and improve communication about the importance of single cell human cell atlases, particularly for the placenta women. Nigeria, don't forget, remains the country with the largest or highest maternal mortality globally. So this particular project brings to bear an opportunity to actually start to tackle the problem via a scientific perspective. And so Musa worked on developing the community engagement plan, on enlightenment, recruitment, and even planning healthcare plans for pregnant women who will be enrolled in the study. And once Musa is able to actually successfully enroll these patients or these donors, placental donors, in his study, in his locality in Kaduna State, these placental samples, when collected, will be shipped, which is about an hour's drive from Kaduna State to my lab, Idowu Aimola's lab at Ahmadu Bello University in Zaria, where the single cell libraries for the placentas will then be generated using the 10x pipeline. And we hope that once this is actually generated, once this single cell atlas is generated, they will then be shipped to the United States to Dr. Benedict's lab, the computational biologist, where they will be sequenced, and then the data will be well analyzed. So in trying to map out the placenta cell, the placenta tissue from African ancestry, we have decided to actually look at different regions, most of the major regions, including the chorionic plate, the chorionic villi, the decidual tissue, basal plate, and some from the umbilical cord. And we probably will also profile maternal blood. And then this, like I said, will be barcoded and single-cell libraries generated at Ahmadu Bello University. This is all trying to build up on our individual strengths. We targeted 3 different ethnicities in the initial stage. And what we try to do is we try to get the dynamic nature of pregnancy to actually capture the process in a single cell form, the process of pregnancy, right from the first trimester down to the 3rd trimester. And so that we can actually scale up this project eventually, and with additional collaborations and bringing in other additional expertise to actually effectively look into the data. But we're confident that our team right now is well placed and well versed with the required expertise in computational biology, community engagement, and cell biology to actually generate novel placental single cell data from Nigerian women pregnant population. Musa, you go ahead.
In the initial phase, we had a period in which we were building trust. So at that time, we were studying one another's personality and motivations. So it was really a hectic period because we had to talk sometimes over like 8 hours in the night, trying to sort out very minute details of what we intend to do. So during that period, we now moved to a phase in which we did a lot of negotiation about who was going to do this and who was going to do that. And it was thereafter that we began to take joint action and our creativity began to be expressed. Later on, we began to solve problems because now that we have built trust and we have really known who we are and what we all are contributing to the project, we also began to bring the structure that we needed to work together. And during that time, we also began to share knowledge because if we're talking about how to prepare cell libraries, Dr. Idowu, whose entire period in NHGRI was focused on the single cell research, he will be the go-to person to tell us, okay, these are the techniques that we needed to employ for specific problems that we are beginning to foresee. Now, if we come backwards to the beginning, because how we decided to work was more or less like a relay team. So I am the one that is starting the race, and we know that it is critical that for every member of the relay team to do his or her own bit very well so that we will successfully get to our goal. So with that, we shared a lot of knowledge and we created a lot of value, which we now eventually saw as winning the grant. And now that we're back in Africa, we have begun to set up all the infrastructure that we need to get our project going. Now, all this has happened not without challenges. And what kind of challenges am I talking about? Firstly, we had a fiscal constraint because Dr. Anchang happens to be in Research Triangle Park in North Carolina, USA. Initially, it wasn't a problem because I saw him down the hall from my room in our lab in our EHS. So, but when I moved back to Africa last December, now that fiscal constraint became an issue. Although the last 2 years with COVID-19, that fiscal constraint has been an issue, but we're able to work it out because we were all in the same time zone. So now moving back to Africa, initially, Dr. Anchang was 6 hours behind us. Now it's 5 hours. So sometimes, getting the appropriate time to meet could be really challenging. But since we were able to distribute tasks, we were able to go around that challenge. Now, there is also the problem of real-time discussion because sometimes he may wake up in the morning, he may want to get in touch with us, but morning is actually evening for us. So he has to leave a message, or we have to leave a message for him, in our morning. Meanwhile, it is night in the Eastern Daytime in the United States. So later when he wakes up, he gives us a reply. Then there's a operational problem. There is a security concern in sharing information on public server. He works for a United States government entity, the NIH. We work for universities in Africa. So that is also a very big challenge. So, but before we left for Africa, using the resources of NIH, we were able to overcome that challenge to get a common place where we could deposit our work and share it. Then there's operational problem. There are security concerns, of course, about sharing information on public server, which I had mentioned is also a physical constraint. So, we were able to overcome that, and we are doing more to be sure that in the future, we don't run into any problem that will compromise our work. Now, auditing and compliance issue in developmental management is also a very big problem, because Dr. Idowu and I are in different institutions. Dr. Anchan works in NIH. So the compliance issues that we anticipate, we really discussed it, and we make a lot of consultation within our organization so that we don't run into any problem, especially that there is going to be material transfer between the institutions and also data transfer. So these are issues that, we needed to do a lot of consultation with our institutions so that we know the appropriate thing to do, and that is what we are doing. Now, good teamwork. The demand of understanding and sharing codes written by others. In the future, we anticipate that, we're going to be sharing codes. So far, we haven't gotten to that real challenge, but it's about writing, certain documents. Sometimes I share with them, or Dr. Idowu writing some documents, that is supposed to be circulated amongst us. So because of our unique backgrounds, sometimes we need to explain, in more detail what we really mean in the documents that we share. So in the future, we expect that we're going to be sharing codes between the different groups. And now, if we are to maintain a good teamwork, we ought to have a very good understanding of what we are doing. Then good documentation is required for collaboration, and it's difficult to make, because when we hold meetings, sometimes the meeting runs into hours. There has to be minutes taken, and what are the action points. So I was appointed as the secretary of the group, so I keep minutes of our meeting, and sometimes the discussions could be very intense, and so I have to make very good notes so that we clearly know what issue we're talking about, and if we're to take action in a particular point, it has to be agreed by all the parties involved. So basically, these are challenges that we have been facing, and over time we've been working towards overcoming those challenges. So in the future, what is really our goal? Because now that we've come together and we're going to execute this particular project, we expect that we are going to build a very good team. Okay, so our long-term goal is that for 5 to 10 years in which we plan to be working together, and into the future, we plan to be supporting one another. We want to also carry out more team work. And in this way, we are going to be sharing students who are going to be trained in the various labs. So for example, Dr. Idowu and I have our co-supervising PhD candidates that will be coming to my lab, and also candidates from my own lab will be going to his lab. One major capacity building effort that we are doing, which is what I described, is that we will also recruit people with backgrounds in mathematics and computer science, so that they can also be trained in computational biology, so that in the future we will have independent capacities in different labs to have people with those skills, so that we can get our work well done. So communication is very key to our collaboration. So we will be doing a lot of communication efforts in the coming months, so as to make people within our group and also the society that we're serving to know what we're doing. And this will engender more trust, and a lot of younger people who we expect will be motivated to want to join our group, so as to be part of the single cell research effort in Africa. So we want to be inspire future scientists with what we're doing, so that they can succeed and we all succeed. As it is said, team, T-E-A-M, together each achieves more. So we want to thank all of you for the attention that you gave us.
I wanted to seize the opportunity to thank everyone and the organizers of this meeting to give me the platform to share a bit about what we've been doing on the context of Africa, and then also how we've been able to use genomics to address some very critical health challenges, especially in Africa, and then looking forward and how we could leverage the existing platform actually to move what we've done. I mean, talking about genomics to contribute significantly to the human cell atlas, and then to basically just do single cell sequencing and the single cell genomics. So I mean, the HCA is giving me these titles: the growing of the African genomics, the African genome, and then lessons for ACA. So Christian Happi is my name. I'm the director of the African Center of Excellence for Genomic of Infectious Disease at Redimus University. And then our center is a WHO and Africa designated reference and continental reference center for infectious disease genomic research and innovation in Africa. We're also a reference lab for the World Bank, and we are here at Redimus University. So, and what I will be talking today is more or less about how we've been able genomic to transform outbreak response in Africa, and then how much this could be leveraged in order to move HCA and especially single cell genomic, I mean, in the African continent. So and in order to do that, I will basically just really start with the premise that everyone knows here. So the genomic revolution and then the technologies that are accruing from that have revolutionized the way healthcare and research is being conducted. But unfortunately, many of these technological and health benefit and economical benefit from the genomic revolution have really fully yet, have yet fully, simply because, there is really no know-how on the ground from African scientists, or enough know-how from African scientists, and also and that really hampers, the development and then the deployment of these technologies. So a few years back, Africa, for instance, I would say that, what was really important for us is basically to see how we can harness the knowledge and the technology, accruing from genomics to address problems. And what are those problems that we think are very peculiar, to Africa and very pressing for the African continent? It's actually the one of infectious disease. One of the speakers, Idowu was referring to how this could be, extended to infectious disease. And then just looking at this map here, you could have a sense of what it is in terms of, understanding, how the burden of infectious disease. These are diseases. I mean, here, if you look at this on this map, you will see that these are diseases with pandemic. And then if you take out COVID and then probably SARS, you're going to realize that the majority of the diseases that have the pandemic or epidemic potential emerge from Africa, and then probably, and Africa, being very rich in its own biodiversity, definitely is a reason why the continent has a lot of these viruses, and we should definitely leverage, these pathogens in order to start developing novel technologies and novel approaches for, epidemic or pandemic preemption and countermeasures in order to contain, future epidemics. So in order to do that, a few years ago, we set up a platform which is called the African Center of Excellence for Genomic of Infectious Diseases, with resources from both the World Bank and then the USNIH. And this platform is basically built around around clinical testing centers in Africa, and then academic institutions, and then that have been working together for a long time of research and then capacity building. And this within Africa, within Nigeria, Sierra Leone, and then Senegal in West Africa, but also have international partners like Harvard University and then the Broad Institute, and then Illumina, and then Cogenics. And this list has really expanded since the beginning of this consortium, expanded to something that is pretty much global now. So, and the mission for this consortium that I've set up, or the center that I put up here, are twofold. One is educational, and then the second one is research. So the first, which is the educational, mission, and that is many bank, is really to, first of all, create, vibrant, academic and research environment that transcends national boundaries. The reason why we want to create an environment that transcends national boundaries is simply because we recognize the fact that in Africa, there's no brain circulation, knowledge cage, and all because of the colonial, what I call immigration rules inherited. But we really want to create an environment where young Africans could come and then conduct relevant, responsive, and ethically highly translational genomic-based research for health development on the continent. And in order to do that, we really feel that it's very important, first of all, to build that critical mass of young African with very good knowledge, in the face of in genomics, but then and then also train them on how they can use or empower them on how they can use this knowledge, the genomic knowledge, to develop, genomic-based tools toward prevention and elimination of infectious diseases. But however, in order to really empower them, we needed to create, genomic curricula, because we're bringing something new in the field and new in the academic, landscape of Africa. And we set up, we generated, new curricula in molecular biology, also in bioinformatics. And I'm very glad to say that these curricula that we set up have all been internationally accredited. And that really give them the pedigree that they will be needing at the end of the day or at the end of the training in order to, to use this knowledge and in order to translate done into very tangible impact in the field. But in addition to do that, we also usually do community engagement toward prevention and then public health education. So, and then the research that we this whole education platform is really driven by research because it's at postgraduate level, and focusing on something that is important, which is, what I call identifying the drivers of febrile illness, on the African continent. But then what I would try to say here is basically just ultimately our goal is actually to build the next generation of African pathogen hunters using genomics as a way to lead the charge against infectious disease and developing countermeasures for the world. But then I will just take a few use cases to demonstrate how we've been able to use and harness genomic knowledge to actually transform what we call outbreak response, not only to Africa, but actually have a significant impact on outbreak response in the world. This really started, in 2014 when we had the Ebola outbreak in West Africa. And what we did at the time, and that was unconventional, was actually start using genomic sequences as a way to, do real-time genomic sequence, generate data toward, helping the public health system to respond to an epidemic. And that was the first time this was being done. And what we did that was unprecedented was the fact that, within the first 3 weeks of the outbreak, we generated the first 99 full genome of Ebola in the midst of an outbreak, and then made this information publicly available, share with the international research community so that they could use, develop, countermeasures, vaccine therapies, and then diagnostics. And this really set up, what I call the framework that we're using today to respond to COVID, for instance. So we actually pioneered open data sharing and open data access during and we're very glad today that that was embraced. And then today everybody's talking about GISET, where we're all sharing data, where the global community is sharing data, and then these data are being used, to respond to the epidemic. Very glad to say that we pioneered this in 2014 during the Ebola outbreak in West Africa, which was the largest Ebola outbreak ever. But in addition to that, we set up, what I call, diagnostics technologies and then sequencing methods that we actually made also available, publish this as an SOP so that people could use in order to perform real-time genomic sequencing wherever they were. And then this also provided an opportunity for, technological innovation to see between sample collection and then when we were submitting data in GenBank 10 days. That was a revolution at the time. And then we actually refined it a bit further. You're going to see that afterward. And sharing this data, generating data and putting them in GenBank and then releasing the data and in 10 days was really, impressive. And then we were doing this in order to get fame because our papers, publications, from the data that we made public could only come out about 6 months, 8 months later. So really, I mean, sharing the whole idea that data sharing could be very valuable toward, pioneering the idea that data sharing could be very valuable to an outbreak response. We were able to use such information, as you see here, to show that in the case of Sierra Leone that the outbreak or the epidemic in that country was due to human-to-human transmission, sustained human-to-human transmission, no additional zoonotic introduction from human reservoirs, but then also no newly imported diversity from other countries. And then this is exactly what is being done now. Using genomic data, you could see here, for instance, the case of Sierra Leone in 2014, seeing how we have different variant of the Ebola in Sierra Leone, and then around that June, one of the variant actually started taking over. You could see that here, and then this again was during an outbreak, how you could use genomics as a way to show emergence of new variant and then how those variants actually get spread. But apart from that, we took it a step further, demonstrating, that the taking over or the what I call the high transmissibility of that variant was actually due to accumulation of mutation across the virus. Time, such insight could be provided into the virus during an epidemic, and that really was very important. And we're glad that this has become the standard. We're talking about all the different variants: the alpha, the beta, the eta, the gamma, and then the deltas, and then the omicron. Really emerged from pioneer work that we started in. But apart from that, we were able to use this information actually to identify using genomics, viral genomics as a way to identify factors that were very important in spreading and sustaining this outbreak. Here, for instance, just using genome data in time and in space, how we're able to show transmission from person to person, from county to county, district to district, countries to countries, department, and countries to country. How using this to show transmission and then hotspot, and then use such information to help public health respond. But apart from doing this, only just use this to actually help public health respond, we also translated this information into things that were very important in curbing the outbreak in the country. In West African countries. The first thing is that, yes, we generated the genomes, but we also realized that along the line, it was very difficult, continue to testing on PCR long time to actually do conformity diagnosis. And people that left their home with a simple malaria, not Ebola virus disease, going because they had fever, going to Ebola testing center, actually contracted Ebola over there and then took the virus back to their homes. So we realized that, developing more accurate and rapid diagnostics is critical in order to curb this outbreak. And what we did was basically translating that knowledge into the genomic sequence data, into point of care and point of need diagnostics. And we were able to, with colleagues and collaborators, develop the first Ebola rapid diagnostic test, that was approved by the WHO and then the emergency use authorizations. And that really, the introduction of this test around 2015 became really that very major turning point in order to contain this outbreak, because the doctors and then the clinicians and the diagnosticians could have the result of testing in near real time. And that became very important in order to really move this forward. So the second use case, we've been able to use genomics and then show the power of genomics, to address a major health problem in Africa. So Lassa fever is a disease that was discovered in Nigeria in 1969, or described the first time in Nigeria in 1969, that is responsible for viral hemorrhagic fevers. And since, all this while, up to 1987 or 88, there was no molecular diagnostics on the ground. And if you look at it critically, often anytime there was outbreak in Nigeria, samples get transferred to Europe, and then 90% of the people are dead by the time the report comes in a month after. So we set up the very first molecular Lassa fever, in Nigeria, and then this is a disease that kills thousands of people in West Africa every year. But then we set up the first molecular diagnostics, and then apart from setting up the first molecular diagnostic, we actually generated the first, whole genome sequence of this virus. And this really became important. Actually, it was the very first demonstration of using unbiased sequencing to actually show, the viral sequence of a virus creating havoc in a whole part of West Africa. You could see here, between here and then, that this is where the disease covers across West Africa. But apart from sequencing, we're able to demonstrate one thing, that this virus that was really in Africa, or described the first time in Nigeria, in 1969, is actually a much more older virus. That this virus was that emerged about 1,060 years ago from present-day Nigeria, was circulating within Nigeria for 600 years and moved out of Nigeria to go toward West Africa and to other West African countries. But then apart from doing this, we're able to basically just translate this again, using the viral genome sequence data. Since we created the largest, Lassa fever virus genome repository in the world, we're able to develop, another point of care diagnostics, which is the pan-Lassa, because we know that this virus is very diverse. You have different variants or clades, and we're able to create a rapid diagnostic that can capture all of the major variants circulating in West Africa, clade 1 to 4. And this really became also very important for the virus, because apart from just doing this, we were able to use this information, to help, accelerate vaccine development. You can imagine between 2015 and 2020, when we actually, between 2015 when we developed, when we provided the first genome sequence, of the virus, and 2020, we went from a disease that was highly neglected to a disease that had major vaccines that are undergoing clinical trial in West Africa. So again, showing the firepower behind this technology, in accelerating, development of countermeasures. Yeah, we had this publishing cell at that time, got the cover of cell, but really what is important about this is the fact that, we were able to leverage it to understand exactly what were the drivers of resistance and susceptibility to this virus. Because what we observe in the course of our studies is that different people could be exposed to the virus, but only few of them go on to develop the disease. Then the question was, what is really driving, susceptibility or resistance in different African population? We went on to actually do additional studies and realize that, what is important about this is the fact that we identify some genes that are important, in protecting specific populations, due to Lassa fever. But apart from that, we actually, adopted recently, leverage, the gene editing technology, CRISPR, technology in order to develop the first CRISPR, diagnostics for infectious disease. And then this was really published in Nature. And then we have Amy Maxson from Nature that were embedded in our lab for a week. And you could see structures of, our lab here, getting people how we got this technology, developed. And then this really is a very powerful technology that is cheaper, and then that can actually help you, detect this virus, without any tool. Just basically just having this on the paper strip and then get the result in a few minutes. And that helps in the sense that, it's well adapted for a country, for a continent like Africa. But as I said before, so we really went on.
I'm sorry, we need to wrap up the session in just a minute. I apologize.
Right, as I said before, we went on to identify the gene that is responsible for susceptibility or resistance to Lassa fever by scanning the genomes, but also contributed human, to the 1,000 Genome Project, and then the population that we actually work on, which were the Mendes of Sierra Leone, the Ishans of Nigeria, and then the Yorubas of Nigeria seem to be the most diverse. And this diversity is actually driven by infectious disease. But again, just showing here the case whereby you use metagenomic discover 2 new viruses in Nigeria, using another case here where we basically just use genomics as a way to identify an outbreak that is unknown to public health authorities, identify a new yellow fever, strain that has not been circulating in Nigeria for the past 94 years, made the information available to public health authorities, they declare an outbreak, and then they contain that. 10 days. And then for COVID, for instance, we generated the first COVID sequencing in Africa within 48 hours of receiving the samples. But then demonstrating that you could use this as a way to show, track the virus as it goes. You could see here virus tracked over time. But then, developing novel diagnostics, multiplexing that diagnostics, but also to set up a very great platform. Here you could see we've trained about 32 different African countries and sequenced samples from those countries during the outbreak using genomic sequencing. And that explained the reason why you see a sharp increase in the number of African genomes, over time. You could see that here. But then it's really also built on the platform that we have from NovaSeq that to the PacBio, SQL 2E, to all these automation platform that we have, for extraction, for library constructions and everyone. And then this really could be very used. So overall in Africa, you realize that there's over 200,000 genomic platforms, and these are really things that the SGA could actually move this agenda much more forward. You could see them here. So, and then that really becomes exactly what we think, that we can work together, with others to expand the scientific capability of Africa in order to enable sustainable, independent, not only surveillance for outbreak, but also, things like single cell genomics. And then my funders for making this available.